Welcome students to the another lecture. Today we'll discuss about the thin layer chromatography, the basic separation techniques into the sub-factors of the chromatography. In this chromatography, generally the separation is based on the solvent, that is the mobile phase we can call, and the stationary phase. Okay. Stationary phase, generally it is actually a plate or a strip which is coated with the particular oxide material, that is the silica gel. Thin layer chromatography, that is very similar to the paper chromatography, except what it is, we prepare a thin layer of the inert material, that is mostly the oxides of aluminium, magnesiums, or the silicons, okay, instead of the paper, right? Layer of these particular silica oxides, they are made into the form of a slurry, and which is coated onto a particular flat surface, right? And particularly made up of the glass or the metals. Right? Principle is mostly based on the factor of the adsorptions, okay, rather than the partitions. That means the sample is get adsorbed into the particular oxide inert material. The sample which is having the more affinity with the stationary phase, okay, they travels or they separates slowly. That means they get binds attached with the stationary phase, so they takes the time to separate out. Okay. The sample with the small or the lesser affinity with the stationary phase, they travel faster. That means there is no binding of the sample with the affinity phase. That means they are free to move, and therefore they get separated faster. Right? In case of this thin layer chromatography, we have to follow particular steps. We need a chamber, that is developing chamber we can call. We need a stationary phase and the mobile phase, which are very important. Apply the sample, development of the samples, and then finally the detections. Okay? So these are the 5 basic steps in the order to carry out the TLC analysis. First is what it is very important, that is called the stationary phase. Right? Stationary phase generally made up of, as I told you, the oxides of particular silica gel. We prepare the thin slurry of this. Okay. We use a silica gel of a different type, that is the H. Okay, is without binder. That means when we prepare this silica gel, we need the binder so that it can attach to the flat surface permanently or with this strong effect. Okay, therefore we add the binder. Type G silica gel is actually with the calcium sulfate. It is actually a binder. So we mix the silica gel along with the calcium sulfate. Therefore, the strength of attachment is stronger to the glass or the plastic surface. Okay. Sometimes we can also mix the certain fluorescent indicator in the order to detect the samples. Okay. So mostly the stationary phase we use are alumina, or we use cellulose powder or kieselguhr chip. Okay. So these are the different types of the silica gels or the stationary phase that we use. Okay. So this is what you can see here, the flat surface onto which we have attached the particular inert material, that is our gel. Okay. So this gel get binds to particular flat surface like this with the help of a calcium binder. Okay. This. Okay. So this is what the main structure of a silica gel. Okay. Where the several different silicon molecules along with the oxide, they form a polymer and get attached to the flat surface. Glass plate is another important area. Okay. They mostly of the particular plastics or the glass materials are used of a particular dimensions. We can use any different dimensions, depends on the availability of the chambers or the samples, right? Sometimes we can use the microscopic slide also. Okay, for the small and effortless separation, we need the microscopic slide also. We can use it. Okay, so when we have to prepare a glass plate, okay, either we can pour the gel materials onto the glass simply, or we use the spreading technique. Okay, with the help of a spreader, we can spread the thin layer of a silica gel. So these are what the microscopic slide you can see, and these are the glass plates. Okay. So onto the glass plate, this is what this particular, it is called the spreader. Okay, and this particular area where we fill the silica gel, and then moving these spreaders slowly in the forward direction like this. Okay, So this silica gel will form a thin layer like this. Okay. So thin layer can be produced with the help of a particular instrument that is called the spreader. Okay. So this spreader will helps in creation of the very uniform thin line of a silica gel. Once the gel added on the glass surface, we have to dry that particular phase. So this is called the activation of a plate. Okay. For the activation means the drying of the plate, we generally keep into the oven. Okay, particularly at the 100 degrees centigrade for the 30 minutes. Okay. And after that, we put into the desiccator so that the all remaining moistures can be easily removed. Okay. So removing of the moisture is very important. Otherwise, it will interfere with the sample molecules. Therefore, we need to dry the plates completely so that the separations and the observation or the creation of the spot can be very clear in the crisp, right? So this is, as I told you, this is what the spreader. Okay, and we feed the gel here and then moving into the particular direction, it creates the thin layer of a silica gel onto the plates. Okay. This is what here we put into the desiccator so that all the moisture from the plate can be removed. And this is pouring method of on the microscopic side. We add the slurry, okay, and then rotating the slide so that it create the uniform thin layer on the slide molecule. When we do preparation for the plate, this is what exactly, this is your gel, okay, silica gel mixed with the particular distilled water in the order to make them a slurry. Okay, we call it swelling of a particular silica. So mix it with the mortar, okay, slowly, uniformly, and then pour this preparation of slurry over the slide. Okay, so tilt the slide slowly in the order to move the silica gel along the whole area of a slide. Okay, so keep on rotating the slide so that the uniform thin layer of silicon oxide can be produced over the microscopic slide. Then what we next, we dry that slide into a particular oven here, okay, in the order to remove all the moistures present into the microscopic plate. Okay, therefore they get dried and ready for the separation. So after this is after the drying. Okay, once the slides get dried, okay, they are generally ready for this. This is what is the application of a sample. In this application of a sample, generally we add with the capillary tube or a micropipette. Okay, very small opening of a particular pipettes. Okay, we'll take the very small amount of a sample into this micropipette and then we put onto the glass plate. Then we will add that particular plates or the micropipette slide into a beaker either or on a jar. Okay, this is what the development jar, okay, made up of mostly the glass, or we can use the beaker also sometime, right? So we'll allow the plates along with the sample molecule, okay, will add into the development chamber. Now the bottom of the developing chamber is just filled with the buffer. Okay, so small amount of the glass plate is dipped into the buffer so that they can rise along the capillary reaction. Okay, so this particular bottom portion is filled up with the small amount of the solvent, right? Now mobile phase, which type of mobile phase can be used, it depends on the several factors such as the nature of the substance, okay, whether it is a polar or non-polar. So accordingly we can use the solvent, nature of the stationary phase, the type of chromatography, and the separation. Okay, so generally we use pyridine, petroleum ether, carbon tetrachloride, acetone, water, glycerol, ethanol, or the benzene. Okay, so there are several choices are available for solvent systems. Okay, so this we all use based on the type of substance we have to isolate. Once the isolation is done, we have to retake the samples. Okay, so there are 4 methods: iodine chamber method, sulfuric acid spray method, UV chamber, or the using certain UV dyes. Okay, so iodine chamber, what generally we do, we keep our plate, okay, silica gel plate along with the sample isolated inside the jar which is filled up with the iodine. So iodine reacts with the sample component and then they give the spots like this. Okay, jaha the sample that is isolated, iodine attached to that particular spot. Because of the affinity of iodine with the sample molecule, they reacts and produce a visual colored reactions, right? This is what the sulfuric acid spray. We have to just spray particular acid molecule onto the plate so that the sample can become visualized here like this. And this is the UV chamber, okay, where simply in case of particular fluorescent compound, you have to put this our plate into the UV chamber and turn on the UV light. You will get the fluorescence like this. Okay, so there is the direct detection of the sample based on their fluorescence. So these are what different methods of detecting the samples on the glass plate by the TLC method, right? So in this way, overall things can be done. Prepare the slurry, prepare the plate, put into the jar, okay, which is filled up with the solvent, right? Then solvent, according to their capillary action, they rise, okay, take along with their sample, because the samples is, if soluble into the solvent, they can move like this, okay. Then finally at a particular time they get rich separated, okay, and this separation is particularly calculated by the RF values, okay. And for the detection, in case of a fluorescent molecule, simply we have to incident the UV light, okay. We have to just give the UV light to the plate so that the sample spots can become visible, because they get fluorescence, okay, in case of fluorescence spot. So this is the overall process of TLC. So overall process of TLC, we can see here, okay, a sample, we need a plate and we need a development chamber. Simply add the spot of a sample with the needle, okay, put that plate into a developing chamber, okay, along with the solvent at the bottom. Now this solvent, through the capillary action, they get rise, okay. So the stationary phase is your plate and the mobile phase at the bottom, okay. So the solvent get in upward position, they get rise. Since the sample molecule are soluble, so they, along with the flow of solvent, they also get move, okay, and then separated into the individual form of a different sample components, right? So in this way you can see the sample components are separated, right? In case of fluorescent compound, okay, what then we have to do? We have to put the sample, fluorescent sample, over the strip, okay, then put the whole system under the UV light source, okay. So you can see the fluorescence band under the UV light, which is traveling along with the solvent molecule, okay. So detection, we do not need the further process of detection. Detection is the in live, right? We just need to have the ultraviolet systems through which we can carry out the particular separation. So there, here you can see this, the sample molecules, they are rising, okay, which are fluorescence, and the fluorescence are produced in presence of the UV light. So in this way, the UV light components, UV fluorescence component can be easily isolated. Okay? The application of TLC, mostly we have, we can check the purity of a sample. Okay? Examination of reaction, that is the course of reaction, the chemical reaction, we can identify. Identification of a compound by chemical analysis, mostly we can use it in the pharmaceuticals industry and the food and cosmetic industry. So there is the vast applied area of application of this particular thin layer chromatography, which is a very basic method of the separation with the help of a chromatographic process, right? So this is all for the today. I hope this all understand to you. We will continue in the next topic in the next class. Thank you.